they look the same color as this stage here, and that's called an orthochromatic. So here you still have that dark, dense nucleus that hasn't been spat out yet, and you have that nice pink hemoglobin-filled cytoplasm, all right? So what's left? At that point, you have what we call a reticular network of a little bit of RER that's sitting in there. You can't really see it with your typical stain, all right? So once it gets rid of that nucleus, we call what's left a reticulocyte because it has a little bit of that reticular network left, all right, in it. That will eventually go away once it's been released out into circulation, all right? So all that you've got left at this point is a bag of hemoglobin with a superstructure underneath the plasma membrane of a cytoskeletal superstructure that's keeping the membrane in its shape, okay? Questions about this process? Everybody happy with it? Okay. So here's some cute things to consider. We all are in the adult stage, and we have 20 to 30 trillion RBCs circulating in our cells right about now. Every second, you're releasing about 2.5 million RBCs from your bone marrow. So you're making them at a tremendous rate, all right? They circulate for about 120 days or so, all right? As you can see here, men have a bit more than women because they have a higher muscle mass, therefore a higher oxygen demand. Therefore, they also need a little bit more iron, all right? Although women need a lot of iron because they lose blood, so it evens out in the end. All right, let's see. So how do you get to the end stage? So these things continue to circulate, all right? They take, what, 20 seconds to get around the whole body? That's pretty quick, right? Do their job. They circulate many, many, many times in their life cycle of 120 days. But as they age, they change in certain ways. The first way, that's not actually listed in my handout here, oh well, actually I'm jumping ahead, sorry. Okay, let's go back. So what things can stimulate that production of all of those red blood cells? So I actually mentioned that there were this many, right? It's a tremendous number of red blood cells that you've got, and you're making so many of them every second, right? What stimulates that? Well, if you have blood loss, it's stimulated, you know, your bone marrow is stimulated to step up production of RBCs. Any damage to the bone marrow that occurs, if you go into a high altitude, anyone ever go to Denver for a few days? Some people, the rapid change induces a lot of headaches because they have, you know, a higher oxygen demand. They're not getting it, right? There's less oxygen up there, and it's difficult, right? Exercise increases the oxygen demand. So if you start an exercise regime, you're going to start making more red blood cells to compensate for that increased oxygen demand. Any kind of disease which causes the RBCs to lyse or break apart, all right, will increase that. And any condition in which you have less hemoglobin than normal. So basically, there's hypoxia, all right, which is occurring. So the common factor here is hypoxia, all right? Okay, so let's talk about the end of the life cycle, which I was jumping into. So these things, after about 120 days, they get taken back out of circulation. How does that happen? The first thing that happens is, remember I talked about how they're flexible and how they have to squeeze through those capillaries? Well, after, 120 days or so, they lose some of that plasticity and they can't squeeze through as easily, all right? So one of the places they like to squeeze, that they have to squeeze through, is in the spleen. There's all of this tortuous kind of like a labyrinth that they have to go through in that organ. And if they lose their flexibility, it slows the process down. And if the process of them getting through there is really slow, guess what finds them? Those phagocytes I talked about yesterday, remember I said that they're specialized cells that gobble up other things? Well, there's specialized cells called macrophages that live in the spleen and in other places like the liver and even the bone marrow, and gobble up these ones that are basically stuck. Well, that's one mechanism. The other mechanism by which they recognize that that cell is in fact not just stuck but old is changes that occur in the plasma membrane. Remember the plasma membrane we talked about yesterday having the phospholipid bilayer? One of the common phospholipids is phosphatidylserine. In a red blood cell, it's typically on the inner surface of that lipid bilayer, so it's facing the cytoplasm. As the red blood cell ages, you get more and more phosphatidylserine on the outer leaflet. And that acts as a signal to those macrophages saying, hey, my time's almost up here, all right? So when they get stuck and they're slowed down because of their plasticity, the macrophages and other phagocytes have a really good opportunity to kind of feel them out and see, do you have receptors? Do you have that phosphatidylserine sitting there on your surface telling me you're ready to go, all right? And if so, they phagocytize them. So that's how they are removed, all right? So here's a pretty diagram talking about that. Much nicer than my explanation, probably. So a certain percentage of RBCs actually just lyse in the blood itself, and macrophages that are circulating through organs and through, throughout the actual peripheral blood can come in and engulf the fragments that are left. The other components are metabolized, as talked about here. You can read this as easily as I can, so there's no point in me reading that to you, right? So I remember I mentioned this just a few minutes ago, all right? What controls the production of these RBCs? Well, hormonally, erythropoietin, which is a protein produced in the kidney, right, a little bit in the liver, but mostly kidneys, is produced and released when the blood oxygen is low, all right? Goes to the bone marrow, stimulates that stem cell, right? And the stem cell starts producing more, all right? So it will increase the production, all right? So erythropoietin is one of the crucial, all right, factors that increases RBC production. Testosterone also increases the production and effectiveness of erythropoietin, so it's kind of like a chain reaction. It acts on erythropoietin, and erythropoietin acts on the stem cells in bone marrow. So what happens when things go wrong? Okay, so there's probably a lot more abnormalities than I would have time to discuss here, but just a couple of them I'll hit on. One of the common indicators that something is wrong with the hemoglobin is these things called Heinz bodies. So these are kind of irregularly shaped and sized bodies that are found within the RBCs, and it indicates damaged clumps of hemoglobin, all right? So it can occur from various environmental or hereditary or pathological causes, all right? We won't go into all of those today. But there are certain stains called vital stains that show them up very nicely. In other words, you have to put them on live cells, not fixed dead ones. So the live cell stains will show these little dark bodies here that you see in the cytoplasm called Heinz bodies. And those are basically just clumps of damaged hemoglobin. Another type of RBC pathology is spherocytosis, all right? So what happens here is that there is an abnormality in the formation of an important protein that's crucial to that membrane skeleton, all right, and that's spectrin. Spectrin is present in a lot of different cell types, and it is very well known to be part of the membrane skeleton, but it's a crucial thing for those RBCs to keep that shape. So without the spectrin having its normal structure, it can't form a normal superstructure, and therefore the membrane skeleton is abnormal. And you get these round red blood cells. So how does that affect you? Two ways, two important ways. The first is that you are less efficient at taking up and releasing oxygen, because that round shape is just not as good as the biconcave disc, all right? The biconcave disc allows for faster diffusion in and out. So that round shape is bad for function. It's also bad for getting through those tiny little blood vessels and those tortuous kind of labyrinths in certain organs like liver and spleen, all right? So those rounded cells, those spherocytes as they're called, get stuck basically, and especially in the spleen, all right, which is meant to be a place where you would clean them out and clean out the senescent ones, but even the normal young ones are getting stuck there. And you end up with a really enlarged spleen called splenomegaly, and this is really a gross looking picture. The spleen is normally about this big. So this is just a humongous spleen, splenomegaly. All right, another abnormality would be sickle cell, all right? Basically in sickle cell, what you have is the hemoglobin clumps together to form these long kind of chains within the red blood cell, and it actually deforms the shape of the entire RBC, all right? So that low mag doesn't do much for you. I think this is way cooler. These are some false color SEMs, and you can see how the abnormal hemoglobin has really completely changed the shape of those cells, all right? These things are not plastic. They don't bend well going through the capillaries. Therefore, they tend to get stuck, and people with sickle cell disease tend to get swollen extremities because of the fact that the blood's clogged up in those little vessels, you know. It's not good at all. The only good thing is that if you're in an area where certain things are endemic, certain diseases are endemic, that those pathogens can't live in these cells. So I don't know, that's really a great thing, but all right, so are there questions about any of this stuff? I think we are done.